All right, great. Thank you. Great conference. I want to apologize. Starting out, I couldn't be here the other days. I was at two other meetings, and it's actually exactly the biomedical domain that Michelle just described. And I just want to say that in many of those presentations, these companies that build solutions to manage biomedical information emphasize, and they put the AI on top, right? And everybody talks about AI in different ways, but they emphasize that their ontology foundation and standards foundation. I think it's very important. So we shouldn't be discouraged, but more like encouraged by those developments. Now I'm just going to do something really simple, like just talk about an update, what we did with bioassay ontology. I first want to acknowledge the people who worked on this. So Catherine, Glennie Pascoe, and Nicolette, and Kati and Meek and Michael Sinclair. Some of them are here in the meeting. Helped develop all of this, did all of this work. And there are many previous contributors to this. And then I also want to acknowledge the collaborators that we see here. And I can probably bring a laser pointer up here so I can point things out. Here we go. Annelie from SciBite, Steve Penn, a couple other ones, Vladimir Pistoia Alliance, collaborators, Barry. I show you some of the things we did. Mark Musin and Asia, she's and was contributing in the past. And then I also want to acknowledge all the projects. You can see some of the symbols listed here that helped contribute to the BAO and various grants from the NIH linked to those projects that helped support this project. Very important to acknowledge all of that support. So here's just a quick outline. I was going to talk a little bit about the background of it. How does it work? How do we model assays, and how do we actually then use it in practice? And then what did we do to extend the content and then the release process? And I try to get through it so we can catch up on time. So here are the number of organizations that use BAO. And I just want to show them here to illustrate that it's reasonably widely used and adopted after many years of development, but also saying that we are not funded by those other than those NIH-funded projects, right? So these are not supporting it, but they're using it. So I'm not complaining about it, I'm just pointing it out. So we are really making an effort to guide the organizations that use it and explain why it's built the way it's built. And mostly that's understood, and then people understand, we do it in that particular way, so we can actually serve various different information systems and applications with it, and not just sort of the particular way they may be thinking about it in one organization, right? All right, so, and then there are a bunch of papers, right? I just show them here just to, illustrate that there's also some papers and it's used for a while. And now this presentation is part of an ICBO paper that we submitted. So, and I also want to thank everybody involved to help it and Bill to be the editor, right, and move it through the process, despite some technical challenges with the system. So thank you guys all for that. So here's like a very brief summary that Catherine made that basically illustrate the standard things, right? I don't think we need to really go through it, right? But we want to have a description of those assays and the relevant components, right? We want to have that reasonably structured so we can actually annotate it. We have a representation that's readable, and we can have some basic reasoning. And of course, we have persistent identifiers. If we do, we link to external sources. So you want to have some cross-domain integration. We do some, like I mentioned, limited sort of axioms and reasoning. And if you do a good job, then we can do more advanced querying and error detection and that. So this is the goal of what we are doing. This is like an older slide. I just want to, just very briefly go through how we can think about annotating an assay, right? So just briefly, these assays, what I'm really talking about here is sort of the process of developing small molecule, ultimately drug candidates or chemical probe candidates. Often it starts with some sort of like screen where you test a lot of compounds, like in biological assays. in a biological assay. It could also be done computationally, right? And then the question is, how do we want to analyze this data in a sort of orthogonal way, right? So we therefore organized it in a way so that we have an assay method, or we call it assay design method. It's basically how do we translate a biological perturbation into something we can detect, right? Into some sort of like detectable signal, right? Then how is that signal detected? Like what's the physical detection method here, right? Then what are we actually screening, what is sort of the biological macromolecule or some other biological element that we are actually investigating here? And then how are we actually quantifying the result, right? Which we call endpoint, right? And then what's the assay format? So is this like a term that is used in the domain? So is it a biochemical assay or cell-based assay or tissue, or is it in some model organism and so on, right? So this is like roughly, I think, the important things we need to recognize. So the goal here is to have this orthogonal annotation of assays, enable this orthogonality so that we can find different or compatible data by organizing by asking different questions depending on what we are interested in, right? And if you're thinking about a high throughput screening campaign, right, then often because you want to avoid artifacts, right, then you confirm the assay with another assay that's orthogonal, right, where the detection doesn't work in the same way, right? After, like, of course, confirming the high throughput data, right? So just to illustrate that, right? So here is an example of a bunch of luciferase assays, right? So kinase enzyme activity, right? ADP glow or something, right? Uses luciferase reporter gene assay, of course, right? Or the actual enzyme activity, right? Or cell viability, it's ATP glow assay, right? So all of those have these design methods, we call them, right? And then we define them, depending on what they use, and they all use the luciferase. Of course, they're all based on chemoluminescence, right? And they all use the luciferin 4 monooxygenase and ATP in some way, right? And now by doing that, these assays are more formally described, so we can ask a simple question. If you just look at a bunch of data from PubChem, and this is a really old example, but I just want to illustrate it. Reporter gene assays, cell viability, luciferase assays, kinase activity, but also like toxicity assay works the same way, like to detecting some P450, right? And then here basically see the compounds, and then we see how promiscuous are they, right? In this particular category of assay. So how many times was that assay, was a compound considered active versus not, right? And if this is one, then it's active everywhere, right? And if it's like green, then it's active like only in a few assays, right? That same compound of that same category, right? And then we can just make a simple index, which is shown here. So now if you look at this, right, we can just simply, by just clustering this, we can look at two different categories, right? One, they're all active in this reporter gene assay, right? And this is active in the luciferase inhibition assay, not in a viability assay, right? And then in some other ones that use the luciferase readout, right? And then we just look at those and we can see those compounds, they are luciferase enzyme stabilizers, right? Enzyme inhibitors, right? So now we know these are sort of artifacts and we know sort of how they work. And then the ones that are not active in those, but they're active in those viability assays, right? Then we know they're generally cytotoxic compounds that you can see here, right? Intercalator or just a reactive compound, right? So this is just a super simple example how it can be useful to organize the data, right? So, all right. So I want to now quickly say say a little bit about how we use it, how it's annotated. So my friends, Barry Buchanan at Collaborative Drug Discovery, and we contributed to this, built this Bioassay Express Bioharmony annotator. This is open, so you can go to bioassayexpress.com, and you see like an interface here, and essentially, if you just look at an example, here's the text, right? And then this annotation tool, this software would essentially suggest a bunch of terms, right? That then a human could confirm or not confirm, right? And this has a simple AI behind it and uses some of the semantics of the ontology. And then this helps a lot to annotate the assays at scale. And then this technology was also used, I believe, in one of the Pistoia assay annotation pilot projects, and I think it's used in a couple of companies to actually annotate their assays.
So this, you can look at a simple workflow. I just have illustrated this here. Of course, I hope you guys can see it, but basically the first step, annotating it, right? Together with some technology that we built on HoloBridge, but it's also integrated here internally. Then we can do a term request and determine a label for it and the description, right? Then this gets reviewed, approved, and then the annotation can happen. But the annotation can already happen here before the term is approved, so that we don't have to interrupt the process, if something new is needed. And they have built a really nice process where you have, different roles, administrators in an organization who can approve that. And while an annotation can still go on at the same time, that then that term gets internally replaced and the URL, the identifier is then added to that system, to this internal data representation system. And here we have, if you go to annotated assays in Bioassay Express, you see a bunch of PubChem assays. I think there are like 3,860 that we have annotated using that technology. And then also quantified how much more efficient it is. So that is part of the CDD world. I'm just giving a little shout out here. It's a lot of functionality, from registration to making predictions via deep learning, and even generating, I think, compounds, analyzing the assay results and so on. And then this part, the assay annotations and ontologies, is part of it as well, to again have the data organized well, right? And this is integrated into the CDD tool suite. So allows to build protocols with fields and ontology terms, and then those are very structured and organized. And here our little bioassay ontology is in there, right, in their tool suite. So I'm happy that they were able to use this, and so just highlighting it because they built this great annotation tool. All right, so with that, I'm just going to talk a little bit about the content improvements and extensions. So talking about the seed, this is an initiative from the Pistoia Alliance, so semantic enrichment of electronic notebooks, the representation of those, they called it seed. And as the PK/PD and safety pharmacology and toxicology work that we did with them. And then we did a little bit of work to refactor the chemical entity inputs in the Uberon anatomical classifications by just adapting in a better way the class hierarchies in those source ontologies in and do a better integration. And then what is currently going on, the in vitro pharmacology working group, they're working to support FDA submissions. So what you want to do is that replace animal models and then have a set of standard of assays that are important for safety, and standardize all of that. And they're working on this, and we collaborating with them. I think Michael Sinclair's in those meetings, and we're gonna engage some more to potentially help describing those types of assays in a formal way. And then there will then essentially a portfolio of assays and results in FDA submissions that we can formalize in the ontology. And then also, we're working with others, the modeling of perturbations. With Galapagos, there was a lot of personnel turnover, so we need to re-engage with them. And then also the assay kits and reagents, that are offered by these huge contract research organizations and the conglomerates that gobbled up a lot of the other companies like Eurofins, to better describe those. And then if they're linked to the formalized assays, then it will be easier if an assay is run and somebody uses that assay kit to just kind of link the formalized representation directly from using an assay kit. So those are the sort of the things we are working on. So I talk a little bit about it, but I want to first quickly explain, what are we talking about here? Pharmacokinetics, pharmacodynamics. Not everybody might be familiar with this. So if you have a drug, it's convenient to be able to eat the drug, right? Because then you don't have to go get an injection or something like that, or some other maybe more complicated route of administration.
inhalation is not always possible, right, for drugs. But there's, of course, effort there as well, right? And then if you think about eating a drug, right, then we, of course, have to think about absorption, distribution, metabolism, and excretion, right, which is ADME. And those properties of a small molecule, that's why we want to have it want it to be a small molecule, right, that it needs to have properties that actually work in the human body, right? It has to go at least once through the liver, right, and get through the stomach, into the blood, and everything needs to go all the way into the cell and sometimes into the nucleus and still be there, right, in sufficient concentrations. So therefore, these properties are super important, right? So, and if you look at a concentration response profile, just very briefly, right, it just defines the the concentration of half activity, right, when the inhibition is like 50%, simplistically speaking. And, if we make it more active, then that would happen at a lower concentration of the drug, right? So, and this is, of course, highly related to how active the drug is in addition, in combination with what are its PK/PD properties, right? And now generally PK pharmacokinetics is the body's effect on the drug. For example, ADME, like permeability assays, microsome stability assays, volume of distribution, clearance assays, right? And then pharmacodynamic, sort of the drug effects on the body, right? So it's everything, so the potency assay, efficacy, therapeutic index, immunoassay, and so on. So pharmacodynamics is a much broader term. And then if you think about I'm going through this very briefly, I just want to give a very, very quick sort of excerpt of it. The safety pharmacology assays, right, is like what are actually targets and mechanisms that could make it dangerous, right? So for drug-drug interactions, for example, if you inhibit CYP450s, right, then and you take different drugs, right? If one of them inhibits it, then the other drug may not metabolize anymore, and it will have a much different profile than it would have had, in a single drug study, right? And therefore, these combinations could be much higher concentrations and could become toxic and could be dangerous, right? So a bunch of those enzymes and receptors fall under the safety pharmacology. GPCRs, mu-opioid receptors, can be very dangerous, to have too much of that. Beta-adrenergic, enzymes, COX, and so on, like various transporters, ion channels, nuclear receptors, these P450 enzymes, like I mentioned, and other ones, right? So this is sort of what we are testing or what generally is tested in safety pharmacology and toxicology. All right, so just briefly, want to talk to you about, this is now in this ICBO paper, so I'm gonna go through very briefly, but there are some substantial changes we made from the previous version, which was something that we did a couple of years ago with SEED, and had done, but we made this quite a bit different. For example, you're not separating out non-rodent PK and rodent PK and make it one, and reorganize the assays, the pharmacokinetics assay in a different way, because now the pharmacokinetic category would be an orthogonal annotation and not directly under this bioassay category. So again, we have this sort of orthogonal classification of assays and so on. And we added a bunch of other endpoints, to working together with them. These are very specific ones, right? But I think, listening to what Michelle demonstrated, could certainly be more simplified, right? And could be made probably more compatible, more understandable with various other representations. So this is also something we're thinking about it for a while, because we have very specific terms and also very specific object properties. But I do very much appreciate that work that's going on to actually provide more generic sort of representations of it, sort of these design patterns and shortcuts, so that then we have better integration, right? So this is something we are also sort of aware of, and we haven't worked on it extensively, but we are aware of it, and we sort of want to make progress in this area as well. So quickly going through the pharmacodynamic classifications, right? Again, so we reorganize those not under this high-level class, it's under bioassay, but in a different way. For example, the signal transduction assays, or apoptosis assays under toxicity, and so on, so that we again, that we have a more biological classification here.
And these other classifications, this is more like for the drug discovery relevance, that can be orthogonal, right? And then we did a lot of like work on, just making the labels clearer, adding human readable definitions, alternative terms, and this, right? Which is of course important so that it's usable and we find things, right? And then in a similar way for the safety farm assays, in which we added new classes for various safety domains like CNS, cardiac, respiratory, and then we added new assays under the safety pharmacology assay parent, like the HERC, patch clamp, action potential, telemetry, and so on. And then added also like again like a bunch of alternative terms, right, and synonyms and so on. So details are in this paper, but I can also ask some more. I wouldn't consider this all like completely final and perfect, but we working on making this as usable as possible, right? So I think this is what it's probably fair to say. Now here's an example of those very specific object properties, right? That maybe I would get hate for from Michel, but we can probably define them better, right? And then have more compatibility. But as it so happens, right, in those very domain-specific representations, right, this is sort of useful for the sort of domain people that use it. But of course we pay, right, because we then lose some level of semantic interoperability, but we have this great work that this can improve. And here we just see a bunch of axioms, right, that were added. So I can just give you just one really simple example, like this BCRP assay. So BCRP is breast cancer resistance protein, and that is a transporter protein that moves drugs and potentially chemotherapeutics out of the cell, right? So that means now if you have a chemotherapy, right, and this protein BCRP is overexpressed, then the drug could get moved out of those cancer cells, that transported out, and then it, of course, it wouldn't be efficacious anymore. So the drug isn't there where it's supposed to be, right? So therefore, it may make sense to think about those inhibition assays, right? And now, how does it work, right? It's pretty straightforward, right? You have a drug, rosuvastatin here, right? And this is basically sort of, it's not really the substrate because it's not modified, but it's moved in and out. So in that sense, it's a substrate of it, right? And then, of course, the format, it would be cell-based. It's in a cell, right? A human, of course, in vitro. This is the particular cell we are using, right? And this is the ADME classification. Again, these are all orthogonal classifications here, right? And then the design, how do we detect it, right? Is liquid chromatography, just to measure how much of their drug is outside versus inside, right? And this is basically how we use it. And then you see here and not everything is, of course, in bio, right? So those come from different ontologies. We use those terms and we bring them into the ontology. But this is just a simple example of how this is defined, right? So now Catherine, she just constructed like a few really simple like T-box reasoning examples, right? You probably all like very familiar with this. But just to see, do those axioms make sense? Do they work? Do we get errors, right? So not necessarily to provide like a highly valuable orthogonal classifications. We did this in other cases, for drug targets. But here, just to see, does it work, right? And does everything make sense? And is sort of like correct? So here, just to walk you through this very briefly, it's pretty straightforward to understand, right? So we can have those drug absorption assays and permeability assays underneath. And there are a bunch of those assays, CALCO2 permeability, a bunch of them, without going through all of them. And you can see that they all have definitions here, right? Some of them are shown over here. Right, and now you see here cell permeability. We also have an axiom, of course, for that, which is shown here. And but there's nothing here, right? So now we can just infer based on those axiomatic definitions the cell permeability assays, right? So what we see there is, those various assays, except I think they're Yeah, there's one, isn't here, that, yeah, this parallel artificial membrane permeability assay, which is PAMPA, right? So the common term is PAMPA, isn't here because that's not in the cell, right? That's an artificial membrane, so it wouldn't classify here, right? So simple example, just to illustrate you this work of a student, right? And again, the same thing we can do for active efflux assays or interference assays, right? How do we classify those? And again, it's a similar thing, right? We have those transporter substrate assays. This is now a substrate assay, right? There's also a breast cancer receptor substrate assay, right? Not the inhibition one. Then we're looking at something as substrate, right? Meaning, does it get moved out of the cell, right? So not does it inhibit this mechanism, right? And then again, we have those axiomatic definitions. They're pretty simple, right? And then we can classify them as active efflux substrate assays under drug excretion assays, right? So we have again an inferred orthogonal classification, which is shown here, right? Okay, so yeah, I mean, I think this is probably not so difficult to understand for people here. So this is just another like illustration. This is very simple, right? Basically, it just shows how we align those Uberon terms that we had in Bao previously, just all like mumbled under a higher level term from the Uberon, how we now have a better substructure organization here by just bringing in those additional hierarchies, so it's a little bit easier to understand, right? So this is basically what we did, and we did the same with CABI. And then in CABI, it's a little bit more complicated because we have a bunch of terms that are not in CABI, so we're working to incorporate them, but we also now have to, I don't show it here. We have to work then also to in our ontology structure to sort of bring our own sort of chemical entity or material chemical entity terms together with the ones from CABI. And in some cases, it's not so easy to put them into CABI because they may not be clearly defined. There's some proprietary dyes and proprietary reagents that CABI wouldn't like so much because they're not clearly defined. We don't know exactly what it is, but they're still used. So we need to incorporate them somehow. So we have this sort of challenge to basically integrate those pieces together. All right, so this is sort of the work we did. And now very briefly, what we are working on, evolving this pharmacology, safety, intelligence. So this is the IVP work. And very trying to make that useful for regulatory support, right? That I already mentioned earlier. So I don't think I need to talk too much more about it. You will probably hear from us at some point in the future about that work. So yeah, I want to show this, the sort of modular architecture we built. So the reason why we did that is a little bit related to what Michelle just showed, that you have one structure, the sort of our Bao structure that I showed you earlier, right? The assay design method, endpoint, assay format, and so on, right? And we have all these vocabularies, for those different categories, right? Including like object properties, and we put them into like a Bao core file, and then we bring that together with the external ontologies, right? Like Go or CLO or other ones, and then bring that into a more complete representation. These all get imported, right? We don't merge them. They're all like individual files. And then essentially you just need this one, open it up in wherever you are, or go to that. It would be called Bao complete OWL, right? And then it just grabs all these other ones and self-assembles, right? But the reason we did that, this organization is so, because we of course, we got a lot of questions. So why don't we use or why don't we have for example, a bar for upper ontology, right? And we did some work on that. And what it in principle allows this architecture, right? That we could replace those sort of or extend those axioms that are in those modules, which then import all these different vocabularies, so that we could have another structure of the ontology that's more compatible with, for example, the OBO ecosystem, right? Or potentially we'll try that if we have some time to do this, looking at the one that Michelle just presented in particular, as we are trying to do in our research to bring all the biomedical, the clinical data together with research data.
So that is challenging, right? So it'd be really cool to have an ontological framework that makes it a little bit easier. So now we have to see like to which extent that works in practice, right? I mean, in our hands in practice, given like resources, but I like that approach a lot, and I sort of do understand, why this is important to do. So now with that more understanding the sort of modular organization, right? We can also understand how we built it. We were asked to add that, and it makes a lot of sense because probably wasn't very clear how we originally described it. But basically what we do is, we do a lot of data curation, right, in various different templates, which isn't shown here, but we basically, write out labels, definitions, all of that, right, basic axioms and so on. And then all of those things are managed in the Bio database with such a bunch of different tables, for entity info, entity relations, and so on. And then from that Bio database, so this is important to manage all the IDs, right? So you don't have conflicts, you have the proper ID ranges and everything. This is, hard to do in files. So this is all in the relational database. And so Kadi she's also in the meeting and in the acknowledgements did all of that work. And then from there, we have a tool, OntoJog, that we built in-house, that now generates all these ontology files we saw earlier, right? The vocabulary files into module files, and also like generates the external IRIs from all these external ontologies from CLO and Uberon, and Cabby and so on, right? And then for those, use OntoFox, generate those, and then all of those things together sort of self-assemble into the final ontology, right? And this is how it works currently. And we are working to make this more robust and also more flexible, right? But this is how it works. I think it's pretty nice, because it allows us, really, it simplifies the maintenance, and we can do releases like, I don't know, every week or something like that. And if we improve this further, we can probably do it even faster. All right, so this is the how it works, and this is basically the simple process where we get a request, we curate the data, put it in the database, generate all the files, validate it, and then the files go on GitHub, and they go on the bioassay ontology website, and then from there they get picked up into the bioportal and also the EBI ontology lookup service for everybody to use. And this is the how we request things. So use GitHub, or Kadi adventurously gave your email here, but you can basically means you can email us. Also use GitHub if there are bulk requests or other any requests or anything you have, or if there are any problems. So we try to be quite responsive to this. All right, so with that, I'm done, and thanking you guys for the attention.